Welcome back. So today we're going to start talking about mitosis. And you'll hear about reproduction a lot. You'll hear about creating new cells. And in creating new cells or creating new organisms, there's really two types of ways to make new organisms or new cells. You're either going to have mitosis or you are going to have meiosis. So mitosis is what we're talking about this week, and then next week we'll talk about meiosis, kind of to compare and contrast the two. Now, immortal cells can spell trouble. Cell division is in sickness and in health. So when you have these immortal cells, that's not necessarily a good thing. You think immortal, living forever, it can be issues. So when we talk about cells and the life cycle of cells and how long that cells are going to live, a lot of times you'll hear people talk about telomeres. And telomeres are these caps to your chromosomes. It's at the end of your chromosomes. And they're these non-coding sections, these sections of DNA that aren't there to code for a specific protein or anything like that. They're not there to be a set of instructions, but they're there to be a protective cap on the tip of each chromosome. So if you look at this chromosome right here, there is a little tip to it, and that is a telomere. Now, every time a cell divides, the telomere happens to get shorter. So it gets shorter each time it divides, and if too much DNA is lost, the cell may eventually die. So, we're constantly creating new cells. We're using mitosis to divide and multiply to make more and more cells, but over time these cells do age and the telomeres start to be lost, and the DNA gets lost, so there's no more cap to that. And what researchers are starting to find out is that telomeres really play a role in aging. As you get older, the telomeres get shorter. The DNA doesn't have that cap that it once did when you were younger, and that is a topic for discussion, a topic for research that researchers are starting to look into to kind of understand the aging process and how is it that one person might age very quickly and other people look very young at an older age. So here they give you kind of just a basic diagram of if this is DNA right here, in the beginning that you have this long telomere as a cap, and over time it starts to, as you divide and multiply, gets shorter and shorter, and before you know it, then you're starting to break off this piece where you are hitting that coding DNA that is going to have instructions for making proteins or for making a trait or who you are, and that is when the cell is going to die. So with something like Hutchinson-Gilford progeria, we see that there is an issue with the telomeres. So some cells rebuild their telomeres. You're going to have single-celled eukaryotes, gametes as well. All right, so sperm and egg are going to be able to rebuild their telomeres. But telomeres rebuilding and uncontrolled cell division, that can actually mean cancer. So it's not necessarily a good thing to rebuild your telomeres. As your cells get older and the telomeres get shorter and eventually they get so short that the cell dies, that isn't necessarily a bad thing. So if you look at someone with this Hutchinson-Gilford progeria, they have an issue with their telomeres, and it actually leads to them having a shorter than normal telomeres, and they actually appear to age at a very rapid rate. So you see these kids right here, they have the Hutchinson-Gilford progeria, and they age very quickly. You see this is a very young boy. These are the parents of these children, and he is starting to look very, very old at such a young age because he has this telomere disorder. So cell division is this ongoing process in most organisms and their tissues, and disruption to normal cell division can have serious consequences. You can have issues with progeria, issues with cancer. So in eukaryotic cells, a protective section of that DNA called telomeres at the end of each chromosome, it plays a role in keeping track of cell division and getting shorter every time the cell divides, and eventually when that cell gets too old, it dies. So if telomeres become too short, it's going to die, and it's going to kind of keep a balance between old cells and new cells. So some types of cells that rebuild their telomeres with each division can become cancerous over time. Now, when we talk about eukaryotic cells, prokaryotic cells, remember eukaryotic cells are the very simple cells that don't really have organelles to them, and prokaryotic cells are the cells that you probably think of, that you remember when you saw in your high school biology class. On the wall they had an animal cell. Well, that's a eukaryotic cell that's going to have a nucleus and a mitochondria and chloroplasts. All of these things are going to be part of a eukaryotic cells. We call them membrane-bound organelles. They have membranes to them. And prokaryotic cells have a single circular chromosome, so their chromosomes are this circle shape. And in eukaryotic cells, they have these linear chromosomes. And what happens is these linear chromosomes are DNA that's all wound up, and in us, this linear DNA actually continues to wind and wind until we end up getting these long strands that, when they're replicating, they kind of look like an X, and that is our linear DNA. So we have linear DNA. Bacterium and prokaryotic cells are going to have this circular DNA. They also have this other additional DNA called a plasmid in there sometimes, but you don't have to necessarily know about that. But so eukaryotic cells, they have that circular DNA, and prokaryotic cells have that circular DNA, and the eukaryotic cells are going to have that linear DNA that can form what we think of as chromosomes. But both are chromosomes. When we talk about mitosis in humans or in organisms, multicellular organisms, we're typically talking about mitosis as a way to create more cells to repair and to heal. So if you cut yourself, you're going to have to repair that by making more cells. That's going to be through mitosis. But cells, or single cellular organisms, are able to reproduce by splitting in half, kind of the same way that we see it in mitochondria, or in mitosis. So while mitosis is there for making new cells in multicellular organisms, we call it binary fission, where an organism is going to split into two organisms. So a cell creates a duplicate of each chromosome. We're going to call that replication. And the parent cell actually divides and creates two daughter cells. So after the parent cell divides, there's no more parent cell. You have these two new form cells that we call daughter cells. It has nothing to do with gender. That's the name we give it, daughter cells. And this is a form of asexual reproduction. No sex goes on. There's no mixing of genes or anything like that. You're going to have this cell up here, and this cell is going to make a copy. So we have this circular DNA right here, the circular chromosome, and we're going to make a copy of it. And when we make a copy of it, then we're going to have two copies. So here they show you, hey, we have one, we made a copy of it, now we have two, which is this one along with this one, are two copies. One's going to go into one cell, the other's going to go into another cell. So you have this copy here, you have this copy here, and this one's going to go here, this one's going to go here, and now you have two new daughter cells, and that is called binary fission in single-celled organisms. But when it happens in multicellular organisms, we're going to refer to it as mitosis. So in most bacteria and archaea, the genetic information is carried in a single circular chromosome, where we are going to have, or eukaryotes are going to have, the linear chromosome. And you see this because bacteria have less genetic code. We have a lot more genetic code. And bacteria divide by a type of sexual reproduction that we call binary fission. So when we talk about mitosis and talking about the cell cycle, you see that as much as there's the time when the cell does divide, which is called the mitotic phase, or called mitosis, there is a period of time where the cell is really just being a cell, or is really preparing to split or make new cells. So we have this time where we call it the mitotic phase, or mitosis, and then there's also time of just being a cell. So if we have a cell that just split from mitosis, we're going to have this new cell right here. And what it goes through first is the G1 phase. Okay, we call that the gap 1 or the G1 phase. And the cell's primary growth phase is this G1 phase. And normal cell function is taking place. This is just where it's kind of living its life. and it's currently a parent cell, we could say. And then we're going to hit the G0, and that's going to be some cells are going to pause in the G1 phase and enter the state called the G0 or the gap 0 phase, which is a resting phase outside of the cell cycle. And cells may stay here for days or even years. But once we get through that, if we so choose to, otherwise after the G1 phase, you're going to make it to the S phase. So you got G1 goes to the S phase unless it's going to make a segue into the gap 0 phase. So in the S phase, this is S for synthesis, DNA synthesis, making new DNA. This is where the cell begins preparations for division. So as much as in the gap 1 phase it is kind of just being a cell, now it starts to prepare for mitosis. So in the S phase, it's making more DNA so that eventually there are 2 copies of the DNA and they can split. And then when you move from G1 to the S phase onto the gap 2 phase, in the G2 phase, you're going to see that the second period of growth and preparation for cell division is going to occur, and then you are going to go through mitosis and eventually cytokinesis. And what happens is, in mitosis, it has its own phases to it, okay? And what we like to say is I, P, Mat, C. Okay, so you're going to have an interphase, you're going to have a prophase, metaphase, anaphase, telophase, and then you're going to have cytokinesis. So when you're looking at this, it just kind of a abbreviation, or you have your mitotic phase going to your G1 or gap 1 phase, on to your S phase, to your gap 2 phase, and then back to mitosis. And this is constantly occurring in order to make more and more new cells. So in eukaryotic somatic cells, they alternate in a cycle between cell division and other cell activities, just being a cell living. The cell division portion of the cycle is called the mitotic phase, and then you're going to have the other phases where you're going to have your gap 1, your S, your gap 2 phases as well. When you look at the DNA, what happens is you have these complementary base pairings, which makes it possible to produce 2 identical strands by separating the parent molecule and using some strand as a template, each strand as a template to build new ones. So we have this DNA molecule right here, and actually if we split it in half, I can use this one to make a new one and this one to make a new one, because these are made up of these nucleotides. And if I know what nucleotide is on one, I can actually continue to build it by adding new nucleotides. So A is always going to go with T, so I know, hey, that's T. Well, T always goes with A, so I know, hey, that can become A. A goes with T, T goes with A. So now I can start building it, and over time, as you add to it, so this is going to connect with this would connect with an A, you can now create two different, or not different, but two separate identical daughter DNA molecules that you can then split up into 2 new cells. So DNA is actually made up of 3 basic parts. You're going to have a 5-carbon atom or sugar molecule, and you're going to see that in DNA it is deoxyribose. That's the name of the sugar. So DNA stands for deoxyribonucleic acid. Well, deoxyribo in deoxyribonucleic acid comes from this fact that it's made out of this deoxyribose sugar. And then apart from that sugar molecule, you are also going to have this phosphate group, okay? So each new nucleotide can be added only to the OH connected to the 3 prime. All right, so this carbon is numbered as we refer to it as the 3 prime, and then we're also going to have the 5 prime carbon. So we say that a DNA strand has a 5 prime end and a 3 prime end. So here you see that this is the 5 prime, and down here would be the 3 prime, and then they run unilaterally, okay, kind of parallel in the opposite direction, or we call it anti-parallel. And anti-parallel is that this goes from 5 to 3 prime, and this way it goes from 3 to 5, or upside down, 5 to 3. So when we're looking at our DNA molecule, you're going to have your sugar, you're going to have your phosphate group, and then you're also going to have your nucleotides. And these nucleotides, or I should say, are nitrogenous bases. Right, so the whole thing is a nucleotide, and you're going to have these nitrogenous bases, which are our letters. And we know that A is always going to go with T, and T is always going to go with A. C is going to go with G, and G is always going to go with C. So we know our entire code just by looking at half of the DNA. And DNA is this twisted ladder, all right, that we call a double helix. This double helix just means that it's this ladder that is twisted. So in DNA replication, we have to replicate the DNA. If we want mitosis to occur, we want to be able to split up the cell into two new daughter cells, that parent cell into two new daughter cells. In order to do that, well, each one needs their own set of DNA. If you just split everything in half, one's going to have half DNA, the other one's going to have the other half of the DNA. But we want both to have the full amount of DNA so that they can be normal cells. So you're going to have this unwinding and separation, which occurs thanks to DNA helicase. So DNA helicase is able to split the DNA in half, this ladder in half. And what happens then is that you are going to use this DNA polymerase to build the new one, to take these nucleic acid, or I'm sorry, these nitrogenous bases or these nucleotides, and add them to this strand, this template to it. So enzymes connect the appropriate nucleotides to the growing new strand, and the nucleotides are added to the 3' end of the growing new strands. So mutations can occur during replication. Mutations are going to be an error or a change in the DNA or the genetic code or genetic instructions. And there are errors during this, but potentially it sounds like a bad thing, and it can be a bad thing, but potentially it could be a good thing. So every time a cell divides, that cell's DNA must first replicate itself so that each new daughter cell is going to have that. And the way that this occurs is that DNA duplication or DNA replication is going to occur, and it is going to take place because of enzymes such as DNA helicase and DNA polymerase, which are the enzymes that are catalyzing the reaction of splitting up the DNA and adding new nitrogenous bases or new nucleotides to the pre-existing template strand. So the end result is two double-stranded DNA, two full DNA molecules that are identical to each other. All enzymes are going to proofread and repair the DNA during any replication or that duplication process, and some errors may occur. So, you know, an error can lead to a disease or a problem or something that is going to lead to issues that you don't want. Or it can lead to you having some new trait, and a new trait is sometimes good. Evolution, it takes place because certain organisms along the way, they're going to mutate, they're going to change, and sometimes that change is actually better or beneficial or helps you in a changing environment or in a tough environment to live in, and that is what is going to help you to survive in that environment. So now, what is dust? Well, mitosis has two purposes: growth and development. And we see that mitosis is taking place in the tip of roots, and the act of shaving actually damages thousands of cells, which must be replaced. Well, how does that occur? It happens through mitosis. So what is dust? Well, some 90% of dust is just dead skin cells. So you look at dust in your house, you think, oh, it must be dirt, there's stuff in the air. It's really your dead skin cells. So if you ever notice the rooms, if you have any rooms in your house or your apartment that you don't really go in nearly as much as the other rooms, you're going to notice that the dust doesn't build up. Well, the people aren't in there shedding their skin cells, their dead skin cells all the time, and that's why it doesn't get as dusty in there. But if it's a room that you're always going in, it seems like that dust builds up very quickly because you're constantly shedding these dead, dying cells as your skin is making new of them. So what you're doing when you're shaving is, if you shave with a razor, you're getting so close, you're actually taking you're instead of those cells coming off on their own over time, you're actually pushing the process along, and that is going to get rid of those old cells as you're making new cells through mitosis. So mitosis is good. It's going to replace worn out cells. It's good for repair. Anytime you're going to cut yourself or hurt yourself, mitosis is going to rebuild that. Nearly all somatic cells, meaning normal body cells, not sperm or egg, are going to undergo mitosis. Okay, sperm and egg, they're there because of meiosis, which we'll talk about next week. In animals, the rate of mitosis varies for different types of cells. So you have red blood cells, which are replaced every 2 to 4 months. All right, you got 60 to 120 days, maybe 150 days. That is going to be the life cycle of red blood cells. But cells lining the intestines are going to be replaced almost every 3 weeks, or just about 3 weeks every time, just about 3 weeks. So apoptosis is something that occurs where we are going to get rid of certain cells, and this is called cell suicide or programmed cell death, where we are getting rid of any cells that are kind of wonky or that we don't want them or something's up with them. So our body is constantly monitoring what cells are in your body, and the cells are actually programmed to kill themselves so that for the greater good of the entire organism. So mitosis is responsible for generating new identical cells, and it makes it possible for organisms to grow and replace cells that do die over time. So now when we talk about, we talked about the mitotic phase, and then we're going to the G1 phase, the S phase, the G2 phase, back to the mitotic phase. Well, the mitotic phase has its own phase to it. So you have interphase, which is what's going to take place when it's not during mitosis. But then after interphase, you're going to have the mitotic phases, where you're going to have prophase, metaphase, anaphase, and telophase. So in prophase, chromosomes are going to condense. Then you're going to see that these chromosomes, once they are condensed and they're kind of all organized in nice little packages that look like X's, you're going to see that in metaphase, the chromosomes are going to line up in the center of the cell. And once that, each chromosome is going to be pulled away from the center or apart during anaphase. And then lastly, you're going to have telophase, where new nuclear membranes are going to form around each complete set of chromosomes, and the cells are going to finally divide and become separate during cytokinesis. So before mitosis begins, in our S phase, you're going to see chromosomes are going to replicate. Sister chromatids are going to condense. So you have you have these identical types of chromosomes, where when we are making things like sperm and egg, we're going to, you have 36 chromosomes, I'm sorry, 46 chromosomes, and you're going to get 23 from mom, 23 and dad from dad. Well, we break them up into your number 1, your number 2, your number 3, all the way up to your 23rd chromosome. And you get one number 1 from your mom, one number 1 from your dad, one number 2 from your mom, one number 2 from your dad. And these homologous chromosomes are going to be the same. So we see that chromosomes during all of the cell cycle, except mitosis, eukaryotic chromosomes are uncoiled and spread out in a diffuse way. Now, during replication, you have these chromatids, where after replication, each chromosome appears as 2 identical linear DNA molecules held together at the centromere. Okay, so this holds together these sister chromatids, and we can actually eventually, you're going to see the centromere separates, and each of the identical DNA molecules is called a chromatid, and together the two are called sister chromatids. So we have a sister chromatid here, and when you split them, they're just going to be called the chromatids. So as mitosis begins, a spindle forms. The spindle fibers are going to attach to the centromeres, and it's going to pull those sister chromatids to the center of the cell. Now, mitosis is a process by which cells duplicate themselves, very important in making new cells, or really in growth and repair. So we see here in anaphase, the chromosomes replicate in the preparation of mitosis. And then you're going to have your prophase, where the membrane, the nuclear membrane breaks down, the sister chromatids are going to condense, and this spindle is going to form. And then in metaphase, the sister chromatids line up at the center of the cell. Now in anaphase, they are going to move apart from each other. Okay, you have these sister chromatids, and they're going to move apart from each other. And then in telophase, the chromosomes begin to coil as the nuclear membrane reassembles around them. So you're forming this nuclear membrane, and then in cytokinesis, the cytoplasm is divided into two daughter cells, and now you have two identical daughter cells that came from one parent cell. So the ultimate result of mitosis, along with cytokinesis, your prophase, your metaphase, your anaphase, your telophase, up to cytokinesis, is the production of two genetically identical cells, these sister cells that came from one parent cell. So lastly, we talk about cell division going out of control. All right, when that process is constantly going on, and it's uncontrolled, and constant division is going on, constant mitosis is going on, and we can't control it, and it's unrestrained, we call this cancer. And as you know, cancer is not a good thing. So it can cause serious health issues. It's the second leading cause of death in the United States, and it's responsible for 20% of all deaths. So it occurs when the disruption of the DNA interferes with the cell's ability to regulate the cell division. So keep these things in mind as we move forward, that mitosis, and we're going to talk about meiosis. These things are kind of confusing topics, but we're going to continue to talk about these in order to wrap our minds around how do cells reproduce, how do organisms reproduce, and what is it that is going on when they are reproducing and making more, and how is it that this genetic information is coding for or is dictating how we are, what our traits are like, how our proteins are made.